Good morning, everyone. Today we are going to finally look into the role of fat as a storage substance. This is the last part of animal nutrition. And let's do a recap session as always. So before this lesson, we looked at the function of the hepatic portal vein with this main learning objective. And we also looked at these learning objectives, which is from 5S, that states that large molecules are synthesized from smaller basic units. This is actually a definition of assimilation, whereby the absorbed food substances are actually being converted into or synthesized into larger substances, for example, glycogen from glucose. You have bigger proteins from amino acids and lipids from glycerol and fatty acids. We looked at the role of liver in metabolism of glucose and amino acids, but we didn't look at the role of fat as a storage substance. We have mentioned about how urea is actually formed and the breakdown of alcohol that occurs in the liver, and this is actually related to detoxification. Assimilation in liver has many parts, and this actually usually comes up in like section B and C, so it's pretty important. The first main function of the liver is to metabolize glucose, second to metabolize amino acids, and the third is detoxification, which means to actually break down harmful substances and convert it into harmless substances. The main blood vessels that are connected to the liver include the hepatic artery, and this actually carries substances into the liver from the heart. So it carries oxygenated blood towards the liver. And we also have the hepatic portal vein, which carries nutrients and minerals into the liver, which includes glucose, amino acids, water, minerals. And we have hepatic vein, and this carries products away from the liver. So this includes glucose, amino acids, urea, and carbon dioxide. Let's first talk about what happens to glucose in the liver. So glucose can be used in the presence of oxygen to make energy during respiration. And in excess, what happens is that the glucose will be converted to this thing right here called glycogen. You can see that there are hexagons here, and that refers to one glucose molecule, and they're connected into branches. So this is what glycogen looks like. Glycogen is stored in the liver, and it's also stored in muscles. When your blood contains less glucose, lower than that's normal, for example, during puasa time, when you're fasting, there is a hormone called glucagon that's released from the pancreas, and it will be carried into the liver, and this glucagon will convert this one right here, the glycogen, into glucose, and that'll then increase the blood glucose level. But when you have high blood glucose, that means, for example, after having a meal that is rich in carbohydrate, your pancreas will release insulin instead, and this is another hormone. And what happens is actually it happens in the opposite. So insulin converts the glucose back into glycogen to be stored in the liver. Amino acids, it's also pretty similar if you make use of it or if it's in excess. So if you make use of it, it's used to build the cell. So it can build the cell membrane, it can build all the organelles in the cell, it can build up proteins and enzymes and hormones as well, and it is used to also repair the cells. So if you look at this diagram here, this is your amino acid structure. So if you have too much amino acids, you don't store it, but you actually break it down. So this process called deamination will break down that amino acid. So the amino acid will be converted into urea, and this urea will actually be released in the hepatic vein. So the hepatic vein will carry out the urea and transports it into the kidney to be removed during excretion. So if you remember this part here, NH2, this is the amino group, and this is called the carbon skeleton. The amino group is the one that is being converted into urea, and the carbon skeleton will be converted into sugar, and it will be stored in the liver as well. Lastly, detoxification, which is the breakdown of harmful substances into harmless substances. Alcohol, for example, is very harmful and it's broken down inside the liver. And when you have too much alcohol, then your liver can take, then this can lead to liver cirrhosis. Okay, so now about today's learning objective, which is this part, and mainly the role of fat as a storage substance. So what happens to fat after absorption? So after being absorbed from the small intestine, so we are going to go through this part first. Here we have your liver and your hepatic portal vein. Other organs involved is actually the heart. Just to recap here, the heart, this is on the left hand side of the heart, and you have the aorta right here. And you also have the vena cava, which is on the right hand side of the heart, that brings deoxygenated blood back to the heart. The inferior vena cava is actually connected to the hepatic vein, and the aorta right here is actually connected to the hepatic artery. So right here you can see is a structure of the villus. It has the epithelium, which is a single cell layer of single cells called the epithelial cells, and inside is a network of capillary, and also this yellow part here, which is the lacteal. So the red part here is connected, so this capillary is actually connected to the aorta, and this vein right here will be connected to the hepatic portal vein. So oxygen will come in here, and then the oxygenated blood will come out and into the liver. But what happens is that this part right here is going to be rich in nutrients when it reaches the liver. This lacteal is not connected to the blood vessels, but instead into this other network called the lymphatic system. And this lymphatic system is actually connected somewhere at the vein around your neck. So let's look into here first. So we want to talk about fat, right? So the lacteal's job is to absorb fat. And what happens is that the glycerol and the fatty acid will actually recombine to form fat globules. And these fat globules will accumulate in the lacteal, and it will transport it into the vein that I talked about just now. So the lacteal will combine with a vein somewhere in your neck, and that's called the subclavian vein, and that's how fat is actually distributed to all parts of your body. It actually bypasses the liver and doesn't get processed in the liver there. And this subclavian vein is connected to the superior vena cava, so the fat's going to enter here and everywhere, and then it's going to be passed on to all parts of your body. So what happens to the fat in the small intestine? So we're gonna go back and rewind. Fat is actually broken down into fatty acids and glycerol, so this is during digestion. So they're going to be separated first, and then what happens during absorption is that fatty acid will combine with the bile pigments. So the bile pigments will combine with fatty acids to form soluble soaps. This is because fatty acids are actually not soluble in water, so otherwise they cannot get into the, the epithelial cell. That's right. Glycerol and soaps will get absorbed into the epithelium by diffusion, and then glycerol and fatty acid will recombine to form fat globules. These fat globules will be transported from the lymphatic capillaries to the lymphatic vessels in the lymphatic system. So you actually have a lot of different types of systems in your body, and you have many of these lymphatic systems right around your neck area. And these fat globules will actually bypass the liver and enter the circulatory system somewhere here through the subclavian vein. So once it's being transported to all parts of your body, then it can be stored in different parts. Fat can also be used as high energy material, so you can break it down and it has a lot of energy. but it's also being used as insulating or storage substances. It is stored in the dermis, which is under your skin, and also around kidneys. This is a picture of the structure of your skin, and you can see these circle parts here, and that is actually your fat. So that's your fat cell, and their main job is to actually store fat. Fat is good at insulating. This means to prevent the heat from escaping from your body. And in terms of your kidney is so important because it helps to remove all the toxic material. So it carries out excretion, so you want to protect it as well. So there's a layer of fat to actually insulate it and protect it and absorb the shock. So what is important for you to know as well is that you know that sometimes there are pictures like these that you have a fatty heart right here, but you actually don't purposely store fat around the heart. So this only happens when you have too much fat in your diet. Fat is also important as is storage for fat-soluble vitamins like vitamin A and D. Okay, now we're done with animal nutrition for good, and now we're going to move on to topic 4, plant nutrition instead. So I asked you guys to do some exercises and watch some videos on how to define photosynthesis, and that's related to this learning objective. You need to understand that photosynthesis is a fundamental process. Fundamental process means that it has to happen, otherwise the plants will die. By which plants manufacture. Manufacture means to make carbohydrates. And we talked about carbohydrates before. And remember that carbohydrates, there are 3 types. You have monosaccharides, disaccharides, and also polysaccharides. And for photosynthesis in plants, they make glucose first, and that's a monosaccharide. And from raw materials. So what do we mean by that? So photosynthesis is a process, and this is how you define it, and this is how you answer your section C. Photosynthesis is a process by which plants make glucose from raw materials like carbon dioxide and water. The raw materials are only carbon dioxide and water, using light energy. So they use the light energy from the sun, and oxygen is produced as a byproduct. Water is also actually released, but oxygen is kind of really important for the ecosystem as well as the plants. So how do you express this photosynthesis in an equation? This is related to the learning objective for C. So in O levels, you need to express this in words or symbols. So this right here, I'm showing you are just pictures, and they represent the structural formula, I think, of all the molecules that is involved in photosynthesis. You can see here that CO2, and that refers to carbon dioxide. And you have water here, and this crazy part right here is your glucose, and that's oxygen. Okay, so just a fact here that's really important. When you're asked for an equation, if they ask for just an equation, you write down the word equation better, because this is much more straightforward than the symbol equation. But sometimes they want it either in word or symbol. Okay, so the word equation is adequate as it is, but remember you need to also talk about light energy and not just write down energy, otherwise you won't get the mark. So how do we convert this into a word equation? Just write down carbon dioxide plus water, and then you draw an arrow, and then you write down light energy, but don't forget to also write down chlorophyll at the bottom, because without chlorophyll you can't absorb light energy. And then you write down after the arrow, that's glucose plus oxygen, and that's enough for your word equation. But for the symbol equation, this is a bit tough because you need to balance the equation actually. So this comes from the scheme of work for O levels, and it says here, if given as an equation in symbols, it must be balanced. The light energy must also be specified rather than just energy. So this is CO2, you know that very well. So that's plus H2O. And the symbol for Okay, let's move on to this first. So light energy and chlorophyll doesn't have any symbols, so you just write them down. After the arrow, it's C6H12O6 and then plus O2. But if you look over here, it's C6, right? Carbon. There's 6 carbons in this one molecule of glucose. But here we only have 1 carbon. So to balance out this equation, so for example, this is the input and this is the output. How can you have 6 carbons when you put in only 1 carbon in the beginning? So the trick here is just to write down a 6 in front of the CO2 and H2O as well as O2. And that is your balance equation. There is a way for me to calculate this, but I need another video for that. Okay, so the learning objective 4B is for you to learn about how to test for photosynthesis. But I'm only going to explain to you today on how to test for starch, because this is the learning objective, which is to investigate the necessity for chlorophyll, light, and carbon dioxide for photosynthesis using appropriate controls. But to do that, you need to learn this test for starch in the leaf. But why do we test for starch in the leaf in the first place? Okay, we know that photosynthesis occurs if starch is found in the leaves. What happens is that after photosynthesis makes glucose, glucose can either be used up or there's excess. So once glucose is used up, it can be combined with oxygen. And this is during cellular respiration to make energy. And that energy can be used to use up that glucose again to make cellulose cell wall. Otherwise, if you have excess, then it can be stored in the chloroplast, and that's the stroma part. Otherwise, it can be stored in the cytoplasm of the leaf. So this right here is a chloroplast, and I'm going to explain to you how the structure is. And I think it's best if we actually do a recap on the structure of the plant cells. Okay. So what's the structure of the plant cell? You have your cellular cell wall on the outside, and this is the thick layer here. And then on the inside, you have the cell membrane. Inside the cell membrane, it covers or surrounds itself, and enclosed in it is the cytoplasm, which is a jelly-like substance and where all the organelles actually is at. For example, a large central vacuole is really characteristic of a plant cell. Because of the vacuole being large and central, the nucleus would be at the side. It will be pushed to the side. And you have loads of these green colored rice-looking shaped stuff called the chloroplasts. And these are organelles and they carry out photosynthesis. So plant cells are right next to each other, something like this. And actually, we don't really draw this in the diagrams, but you also have mitochondria in plant cells. The mitochondria is found in all types of living cells. The main job is to carry out cell respiration for energy, but we don't usually draw it in plant cells for O levels, but we must know that it's there actually. So here's the structure of the chloroplast, and it's not just green in color, but it has different parts of it. So this chloroplast, if we cut it up, so it actually has 2 layers. It has the outer membrane, and it also has an inner membrane. And inside the inner membrane, it also has its own kind of like a cytoplasm called the stroma. And this stroma, inside it, it has its own organelle called the thylakoid. And this is where chlorophyll is at. So chlorophyll, the green pigments, is actually stored inside this right here. And these guys right here, because it has the chlorophyll pigment, the green color pigment, it actually helps to absorb light energy. And when you have, for example, 6 molecules of water, it will enter the root into the leaf and into the thylakoids. And you also have 6 molecules of carbon dioxide. then all of this will be converted into one molecule of glucose. So this one will actually be first made inside the stroma of the chloroplast, and you have loads of that. So what happens is that the glucose can be made into this part right here, which is the cellulose fiber. And you can see here cellulose is actually made out of this kind of structure where you have this hexagon and then on top of each other in rows, and this is the cellulose fiber. Otherwise, you can even, not you, but the plants can even store it as these two structure called starch. So you can see that it branches out. That is how starch looks like. So starch can be either stored in here in the gray color part, which is the stroma. Otherwise, it can also be stored in the cytoplasm of the plant cell. So that's why we're going to test for starch instead, because we know that the plants are going to make glucose during photosynthesis, and if they are doing a lot of photosynthesis, that means that there will be excess glucose and it will be stored as starch. So how do we test for starch in the leaf? First, you gotta get your potted plant and your pick a leaf as well. Then set up this experiment right here, the apparatus whereby you're going to boil water. So you're going to pick the leaf, make sure to put that leaf in boiling water. And how can you tell if it's boiling? Is if there's bubbles inside the beaker. And you do this for 1 minute. After 1 minute, you turn off the Bunsen burner. Why do we need to boil the leaf? We want to boil the leaf to denature the enzymes. So we want to prevent the enzymes from converting that stored starch back into glucose. Otherwise, we can't test for starch. Okay, next we're going to transfer that leaf into a boiling tube, which is filled with alcohol. And you use the forceps for this. After that, you put this test tube back into the hot water. And allow the heat to bring the alcohol to a boil. Remember that we switched off the heat. It's because alcohol boils at 70 degrees Celsius, whereas water boils at 100. So once you did all the transferring, the temperature would actually go down, even down to 70, and the alcohol will actually boil already. So once it's boiling, what happens is that it's going to remove all of the green pigments. So the chlorophyll is going to come out, but the chloroplast is still going to stay. So it's going to turn white. Now you're going to remove the leaf. It's very brittle, and what you're going to do is remove it out first. Take it out from the hot water, and then you get the forceps, put it back into the water, and make sure that you're going to soften the leaf. The reason to soften the leaf is to make sure that the leaf becomes permeable to iodine solution, which is coming in the next step. So next you get a white tile, get your forceps again, transfer that leaf onto the white tile, and you get iodine solution, and just drop by drop. So step 10, observe for any color changes. So if your observation is that there was a color change seen from yellow brown to blue black, that means that starch is present, and that means that that plant has carried out photosynthesis. But if that leaf remains yellow brown, that means starch was not present. That means that that plant and that leaf didn't carry out photosynthesis. So that's it for today, and hope you guys have a good day.